you hear me? Okay, thank you. Thank you so much, Peter. Wow. Thank you, really. Thank you very much. Since you mentioned the person of anesthesia, I cannot resist actually welcoming my dear friend and colleague, Heinz Kugenberger, who is sitting there. Stand up, stand up. The reason why I say this is the following, and you should all know, because we have to hear all this nonsense from people that have no idea what we are doing. The truth is, a lot of people, most of people I know, scientists, experimentalists, they love animals, they have nothing against animals. Even if you don't love animals and you are indifferent, if you are a scientist, the very first thing you're supposed to think of is the state of the animal during an experiment. Otherwise, you're not investigating what you think you are investigating, right? And the same is true even if the animal is anesthetized. It has to be properly anesthetized. Otherwise, you're wasting your time, whether you love animals or you don't. And I remember very well because in the beginning I was enthusiastic about the vertical magnets that proved to be almost useless for me because there were so many questions that needed to be addressed under anesthesia that basically I hardly use. We usually did some experiments with behaving animals, but not too many. And when I started the surgery, the second and the third time I was trying actually to set up the whole thing with the monkey died after anesthesia, and I was shocked. And I called Unertel, and he sent Heinz. And Heinz, I'll never forget, he was sitting like that, and I was doing the whole preparation of the animal. I did everything, intubation, everything, and so on. And Heinz said to me, there are a lot of things you can improve, but I don't see why you have real trouble. And after he said that, the animal was in this position. I took the tube and I brought it into this position. And he made two big eyes. He said, "You're going to do vertical anesthesia? We need 2 anesthesiologists in the clinic for this." And he took over. Thank you. So I thank you all really wholeheartedly for coming to this symposium. I could not believe my eyes when I saw actually the list of the speakers, of colleagues, of friends. And I am particularly grateful also to the organizing committee that you can see here. And these people have worked more than one can possibly imagine. Conchi, Goran, Hamid, Henri, Oksana, Vishal, and Yushuke over several months in order to organize this wonderful getting together with people that are not just basically audience, but we share a lot, and they are colleagues, and we've been doing work together. So, in an effort to actually try to be okay with the time limitations, and I'm a complete disaster, as many of you know, with basically being okay in time, I'll start with an ultra-short description of my past research and the rationale for pushing the development of the multimodal methodologies that we now use in our institution in Tübingen. And I'll continue with a few words about the neural correlates of the MRI signals, something that we discussed a few times here, and then concentrate on our recent efforts to combine MR imaging with physiology, electrical stimulation, neuropharmacology, and so on and so forth, for the study of so-called brain states and their transitions. Now, here briefly, because you heard already the history, from after my undergraduate studies, I went actually to Munich, to the lab of Ernst Pöppel, and I say that loudly, this was probably one of the most unforgettable periods of my life, 4.5 years there. It was completely wonderful, the whole environment, and also the scientific questions. And then I went to MIT, and then I went to Houston, and then I came to Tübingen. In the first basically case in Munich, in Cambridge, and in Houston, I basically worked with visual system, mainly interested in the beginning in visual perception and object recognition, and also partly in learning and memory. When I came to Tübingen, I decided actually very seriously to try to develop these multimodal methodologies, and the rationale behind this conclusion can be very well demonstrated in a few slides that I'll show you now. Well, people talked about sensory systems, except of the speakers and the senior people. Most of you students or not, you definitely know this standard thing. You have an external stimulus for all sensory systems we have. You have a sensory organ. It could be the eye or the nose or the ear or whatever it is. And usually you project to the sensory and associational thalamus, and then you go to something that people call primary cortex because it's the very first station after the thalamus, and then you go to association neocortex. So far, so good. I'm afraid things are not so simple, though, and there is where the problem starts. The very first thing, the easiest thing, is that you have very strong feedback. And this very strong feedback is undifferentiated. When you go from down to up, things are differentiated, the tangential divisions of cortex are respected, blah, 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 blah. When you go to the feedback connections, they project everywhere, as if they were ignoring basically the specialization of connections in the up mode. And then, as if all that is not enough, you have these wonderful things that are called limbic system and neuromodulatory system. We have quite a few of them that are a true mess for us with the ignorance we have. It's really a puzzle how to understand exactly the modulations, the optimization of SNR, and all these things that these modulatory systems do. You have bidirectional connections between the limbic system and different other basically states of processing in every sensory system, let alone now what is happening in more kind of memory systems and whatever else it is. Now, with on top of it, beyond all this kind of connectivity, you have this not very well understood, probably is phylogenetically preserved because at some point the organisms, they had a sensory and a motor output period without much of processing. And because of that, it turns out basically that in every single station, you have the probability to generate some kind of a pattern that can be used from the motor system. So there is a duplication of every connectivity pattern. If you have two areas that have 2 points that are interconnected, these 2 points, they will be also interconnected through the pulvinar or associational thalamus or whatever it is. So it's quite complicated indeed. In the visual system that we talked about, you know, I don't need to tell you very much. You have basically the eye, and from the eye you go to the lateral geniculate body, and then you go back to the cortex. And identical things you have with the monkeys. The only difference is the monkeys have the fovea somewhere around here, between the lunate and the inferior occipital sulcus. In the humans, they have it back here, most likely because of the huge growth of prefrontal cortex that push things backwards. And then you have also other areas, as we said. I will go very fast because you know what I'm talking about. And you have the same thing in monkeys. And in monkeys, after extensive studies, there are over 40 areas that have visual or almost visual, basically, character. And they have very, very distinct and diverse physiological properties in terms of stimuli that selectively, basically, trigger neural responses. Now, as we said today, in the early areas, you have all these simple features. You start in V1, you have edge orientation, spatial frequency, direction of motion, temporal frequency, and so on and so forth. In the very high areas, you have the story that you hear. There are some cells that suddenly respond to faces, and some other cells respond to hands, which is kind of strange. A single cell has this kind of specialization, but that's what they do. So the question, of course, that comes up is perception and object recognition and all these things do depend just on these neurons. Are these neurons basically the gnostic units, as people used to call them? And not really. So you see here something. How many people here see what is there? Do you see the object that is there? Anybody sees any object there? So these are basically the different pieces of wood, and the fractions are the same and everything, and they have a shading that you see here, but this shading, it makes this appear to be something that from top goes down. Now, what I'm going to do for this very same image, I'll rotate it 180 degrees. Do you see something now? You do, right? Okay. The only thing that changed is basically the shading, all the other things, but suddenly now the structures, they have a different quality, and the object that was hidden in there is emerging, and you very clearly see that there is a deer there. Now, you can see that with all these basically type of figures, the illusory figures that you may have seen, those that may not have seen, you have basically a face here, a dog here, and so on and so forth. Now, the question is, are there neurons responding to such figures only when the figures are perceived? This is the very first thing remember it very vividly, I was traveling from Berkeley to MIT, back still in Peter Schiller's lab. And I was thinking, well, how can I possibly know? And I thought, okay, because these kind of objects that I show you here and the fragmental, let's say, pictures, it's something you cannot very easily use for training an animal. But at some point, I said, okay, what about binocular rivalry? So what's a binocular rivalry? Is something that was invented actually by Wheatstone, that you see here, Sir Charles Wheatstone. And you see here also is the one basically they invented the stereoscope. What he did is he was presenting two different letters within a box, and each one of them presented to one eye, and he noticed that the box basically is absolutely okay, it fuses. Normally you see one box, but the letters alternate. Instead of seeing an A, B that was superimposed, you see only A or B, and this changes over time with teeny periods of superposition of this kind of letters. Now, this binocular rivalry, one of the reasons I did it, I was just looking, trying to find literature about it, and I actually found the thesis work of Pim Leveld, who is sitting here. It's one of the best papers I've ever read. It's a wonderful study, basically going into the details and trying to explain exactly what is happening with the alternation. When you have a particular stimulus, let's say a face against a grating or something, and you're talking about the face, predominance of the face is nothing else but the frequency with which and the time duration with which you have basically the perception of this particular object. So if you increase basically the contrast of a particular stimulus, here, as you see, the contrast is the same, okay? Here, as you see, the contrast is increasing. Then the predominance is increasing. But what Pim noticed, and what's remarkable, is it's increasing in a strange way. The duration of the stimulus remains exactly the same, and the duration of the competing stimulus is reduced over time. So this helped us also very much when we started our first experiments to understand the data that we see with inhibitory neurons and with different kind of things that I don't have time to talk about. Now, this is what I just told you. You see here, you have the stimulus here, and you have here the increasing, basically, contrast. The increasing contrast causes a decrease of predominance of this stimulus, which did not change in properties at all. So, the other thing and last thing, because all this can be used for training correctly animals, is that the phase distribution is basically a gamma distribution. A gamma distribution with a shape parameter of L and size parameter of R is characteristic for sequential Poisson events. It's a very interesting thing. It shows you it's the waiting time for occurrences of different types of events, which means certain things in the nervous system accumulate before you have a true perceptual switch. So we trained monkeys. Actually, the task is very, very easy for training animals. What we did is we have basically the monkeys to see one of these objects. You can see the sunburst here, or you can see the face. And if you show the sunburst, you'll pull the left lever, and it doesn't matter where the stimulus is presented. You can present it to the left eye, you can present it to the right eye, you can present it to both eyes. If he perceives this, he'll pull the left lever. And if he perceives the face, he'll pull the right level. If he perceives the mixture, he learns, and you can check that also with eye movements, then he learns not to touch anything. So he gives you a clear response to the animal after a period of 4 to 6 weeks, and says basically this is the stimulus, and this stimulus clearly perceived, despite of the fact that the natural physical stimulus is not changing at all. okay? So, and here are the behavioral results. You see the perceptual dominance distribution, the gamma distribution, and you see here the humans, what they do with predominance, and what happens basically with this is predominance, and the other thing is basically the probability of switch. And if you see what is happening with monkeys, it's exactly the same thing. So you know that the monkeys do not report random things, but they are very well trained to report exactly what they have been perceiving. Now, we were enthusiastic, and we did a lot of work for many years. And the results, more or less, they showed that you have here, as you see, a very large number of areas that okay. You have a very large number of areas, and as you can see here in the early areas, the percentage of cells that is involved in any kind of perceptual thing is very small. And as you go towards the prefrontal cortex or the temporal lobe or even the parts of parietal lobe, the cells, they fire during the perceptual switches. So the results of all these studies can be summarized in the right, you see at the right side. And what we learned is a large number of neurons exhibit stimulus-specific activity, which is not correlated with any perceptual change. In the early visual areas, perceptual shifts correlate with subtle overall changes, as I had in the firing of neurons. The higher visual areas, like the inferior temporal cortex, PFC, and so on, appear to reflect the resolution. They appear to reflect the resolution of perceptual ambiguity by being mostly active during a perceptual switch. And neural activity related to perceptual changes was reported in association of thalamus, pulvinar, so it's not just cortex or high cortical areas, but there are things in the David Leopold's group. They did some very nice studies. And lesions in basal ganglia appear to induce in humans, also in patients, significant changes in the alternation frequency of multistable perception. So, if you're asking me, no one single visual or quasi-visual area qualifies as the site of perceptual dominance or suppression of a stimulus. Perceptual changes likely reflect system state changes, characterizing all multistable phenomena. But the question then that it came spontaneously in my mind and would come to the mind of anybody here working is, can we ever understand brain function by just studying the responses of single neurons or small microcircuits? If it is so complicated and the whole basically cognitive capacity or each one of the capacities you have is an emergent property of a very complex and large network, what are you going to learn by sitting, let's say, in a particular structure and making 1,000 measurements? So because of that, I actually, said, do we really go in the right direction? And the question whether you can understand perception by studying from neurons is literally like, can we really understand the construction process of an anthill by studying the behavior of small ant groups or even single ants? Well, I don't think so. I don't know if you've ever seen what these anthills are. The internal construction is literally an artwork. I mean, it's amazing what they do. They have the chambers, and you have all these corridors, and they have all this stuff. And people have been studying that using all kinds of mathematical processes to try to understand how can this small population, queens, workers, slaves, but none of them is an ant leader, none of them exactly studied architecture. Each has about 20 internally encoded reactions to stimuli, no control entities, and time dependence in agonistic and antagonistic interactions. So there is a self-organization that you cannot explain so easily from a distance, and there is an emergence phenomenon which characterizes what people call complex dynamic systems. You have a lot of them, as you see here. The upper ones are non-adaptive, relatively easy to model mathematically. The lower ones are adaptive, and they are a complete disaster because we really don't know where to start. So the brain is the ultimate complex dynamic system. As you know very well, it has a huge number of elements, billions of neurons and trillions of connections, high structural complexity, ill-defined elementary units, a nested self-organization, and initial condition-dependent network evolution. The initial condition is a critical thing. It's not just a joke or some kind of a sophisticated statement. There is a very nice metaphor associated with the work with Edward Lorenz that you may know in the fractals and bifurcations, which says the path formation of a tornado can be influenced by the flapping of the wings of a distant butterfly weeks earlier. And the mathematical, basically, description of what Edward was doing supports this because teeny effects completely make it impossible to reproduce a particular behavior or to have the same, basically, model. And now, because of that, oops, what happened here? Okay. Because of that, I actually thought I don't have to just talk about the title of the talk. It was a little bit for fun, just to make it provocative. I don't think I'll ever understand what a brain state in my life. I hope basically the next generations will come up with more solutions. But if you want to approach a state where you can ask the question to begin with, then you need some kind of convergent and interactive things that basically give you data simultaneously at different spatiotemporal scales. And while I thought there is EEG, there is MEG, all that, but MRI, despite its limitations, has an excellent spatial specificity. And with the appropriate methods, and for that I can only thank the firma Bruker, who did an enormous amount of work in the beginning to optimize things for us. And if you have the appropriate hardware, the appropriate pulse sequences, the appropriate software, and everything else that you need, then you can have quite a bit of good spatial resolution, okay temporal resolution, and on top of it, you can have the physiology at the very same time. So, and what I wanted is basically to combine these things and have basically complexity theory and modeling to try to describe the data. In case you're wondering, actually Paul Taggart was known to have written, experiment without theory is blind, but theory without experiment is empty. And it's absolutely true. You need these things together if you want to learn something new. And now, can you do it? Well, with humans, it would be relatively difficult because, as you see here, you have 6 million neurons in a single voxel of a human imaging. You have about 30 billion synapses, 22 kilometer dendrites, and axons 220 kilometers. These are literally calculated. It's not estimation over the whatever. And in monkeys, things are better compared with a very high resolution you can achieve with specialized, basically, things. And on top of it, I'll come to the monkeys, you have another limitation that comes from the volume fraction of the vessels. You have basically natural elements, and the vascular elements are almost 0.3%. 3% means it's very difficult to study exactly the neurovascular coupling and understand exactly what is that you see when you get your imaging data. And in monkeys, things, as I said, are better. You have 120,000 neurons in a voxel, and you have, let's say, 125 microliter. Synapses are 620 million, dendrites, whatever, and axons only 4.5 kilometers. So, again, it's a mess, but at least it's something much more reliable. And of course, we developed the systems. The systems were literally done. It would be impossible to get the systems done without the engagement of the Bruker company. Most of the people, Bruker is one of the few companies, they have really basic research. They were interested, and they helped enormously in trying to develop this. So we have two systems here. And one is 4.7 and the other 7 Tesla, and both were vertical because I said I'm going to have identical conditions that I would have in a normal lab without a magnet. It turned out the questions that we had to ask for 20 years, they hardly allowed 5 or 10% of the experiments to be in alert. Monkeys, most of them were in the anesthetized animals. Here is also the magnets for rats. Now, when the whole thing started working, and it only started working because of people like Axel Oelterman and all the other infrastructure personnel that I have, which were completely ingenious in trying to devise, make a parenthesis. When you're in the magnet and you have an electrode and you alternate the gradients to achieve localization, you induce currents at the level of 50 or 100 amps. And When I started this, everybody told me you are completely wasting your time. Nothing is going to happen. And the only way to deal with that is you try to measure 50 or 100 microvolts, and the induction is 50 volts. It's a joke, right? The only reason it worked, the whole thing, is because the small group I have that we have been working 25 years day and night together, they managed actually successfully to do a noise cancellation interference removal online. There is no A to D converter that can get a signal of, I don't know, 50 volts, and then you try to see the details of microvolts. So you have to remove it before you even start your data analysis. So we did that, and here are the very, very, very first data that were published in 2001. You see here, basically, this is the general signal, broadband signal, up to 35 kilohertz with the new A to D converters. And if you get the root mean square, it gives you an approximation of what is the neural activity changes without telling you specific what kind of activity you see. And if you see the bold, then you get something that looks nice. I mean, it's related to the neural activity, so it's encouraging. And the problem is you have to kind of split the signal. And for those that may not be familiar with this, the splitting of the signal is what I showed you previously. It's called in biophysics mean extracellular field potential. And this mean extracellular field potential captures a lot of things. Usually what we do when we do recordings, traditional recordings, we go close to neurons, we penetrate until we find a neuron next to the tip of the electrode. And if you do that, you get this kind of spikes. You ignore the interspike intervals, you set them to 0. You get basically this line here, everything is 0. It's a point process. And when you have this point process, you can actually say a few things, the neurons are firing or not firing. But if you end up actually having the electrodes fire, which is bad and good, you can see a lot of aspects of the signal, then what happens is you have, let's say, I take one pyramidal neuron, and you see here the dendrites. If the dendrites are depolarized, you have a very strong sink. Current flows into the cell, as you know. And if you have a sink, the Poisson's law in physics says you're going to have somewhere a source, and the source is here. Now, if you have this kind of minus pluses, you have, as you know, a dipole. And the dipole can be shown here, and has different values. And there are two types of things that show also that the physiological signals, to some extent, are limited in their capacities, not only the MRI signals. And there is one arrangement of neurons like the pyramidal cells in cortex where you have basically all the minuses and all the pluses aligned. And you see here all these cells. This is just natural histology, and you see basically the cells are next to each other, so you have an addition of dipoles. But then you have also this arrangement which is called closed field, basically, generator. And in this case, basically, you have the minuses and the pluses in each one of them, as you see here, and they kind of null each other. And if they null each other, basically, the dipole is very small. You see this huge thing here, if I can show you. Where the hell is it? Here. This is a huge dipole, and this is a mini dipole. And these are also large dipoles. So you're going to have always, physiologically also, a little bit of a bias, and you have to take into account where you are, what you expect if you want to interpret 100% the signal. But in all cases, with this method of getting the general mean extracellular field potential, you can generate what people called band-limited power signals that you see here. They were initially known from the EEG studies. If you all know delta, theta, alpha, beta, gamma, and so on and so forth. And there are different, basically, bandwidths. If you take the bandwidth and then you take, basically, you rectify it, you get the absolute value. This absolute value gives you one kind of signal. And what is interesting, and we learned through EEG, this one kind of signal tells you something specific about the brain. There is enough specificity in different frequency ranges. So we used all this information, and here you see the first published, basically, results. You see here the bold signal. You see the spike density function, the MUA. And if you see the LFP is the only one that tells you something about the bold signal that follows up. And if you have basically negative Bolt signal, this negative Bolt signal is always associated with a drop of the multi-unit activity. This is a range of frequency, again, that is above 800 or 900 basically hertz, up to 3,000 or 3,500 hertz, and tells you something about many neurons doing collectively something. So far so good, and what we found with the first observation is that during simple sensory stimulation, Bolt activation is associated with transient MUA, multi-unit activity, and sustained LFP responses with strong initial phasing component. With complex stimuli and during psychophysical tasks, LFP correlates with Bolt, while MUA may often be completely dissociated from the fMRI responses. It was, of course, for us important, if we want to use this for understanding states or understanding other things that we wanted, perception or memory, is why is this happening and under which conditions happening. I'm not going to talk about, I'll talk only about one, but you should know its excitability changes, adaptation of response, its lateral interactions, neuromodulatory effects, feed-forward inhibition, encoding scheme, and so all of them somehow are part of why the MUA very often dissociate from the general, basically, Bolt signal. And the results that we got here, the very first case that I mentioned is excitability changes, and is what I showed you before. You see here very well that there is a very fast adaptation of the signal, and exactly the same thing you see also to behaving animal, absolutely nothing to do with anesthesia. And the second thing that we wanted to do is to try to see neuromodulatory effects. There are many, many studies. I can only mention one of them with serotonin because of the time limitation. What we did, again, was first of all, we did Magnevist injections. If you inject Magnevist, you see it very clearly. And the very first thing that was interesting that can also be relevant to anyone here, even if you don't work with a magnet, is the fact that the speed and the concentration and everything you do during an injection is going to wildly affect basically the correct injection of substance into cortex. And you see here the first one, as you see, it goes all to sub-PL space. So you have to play around with all, basically, the whole things, and if you play around, then actually you can find the correct thing. And after doing that, we also wanted to compare that with an injection of lidocaine. The lidocaine is going to stop activity, you see it here, and if you compare the lidocaine with the Magnevist, you get exactly the same results. And this was basically encouraging for us that what you're going to see, it tells you something indeed about the action of a particular substance on the tissue and on the neural populations. And you see here what happens. We use this one here. It's an agonist, serotonin agonist, and only selectively actually hyperpolarizes the pyramidal cells. It stops the big neurons, and it has no effect on interneurons. And interneurons, they are small guys, but they're extremely expensive. They consume a lot of energy. So changes in the activity of interneurons is dramatically going to change whatever signal you are measuring by taking into account metabolic costs. And you see here that the Bolt is unchanged. Unchanged is also the LFP, and the MUA has dropped, and the Bolt cannot even tell you why it dropped and how it dropped. And of course, here you see also the, in case of visual stimulation, here before the agonist injection, you have normal MUA responses, normal LFP responses, and here after the serotonin agonist injection, you have only LFP and the MUA is flat. And if you see the Bolt, in both cases, you have very strong Bolt. Now, dissociation of MUA and Bolt, as I said, is very, very characteristic and cannot be ignored by anybody who is interested to understand the signals. And we did it for lactate, serotonin, that I said, acetylcholine, and dopamine, and different other studies that actually are still kind of hanging. Now, what did we learn so far? Well, what we learned is the following thing, that you have 4 conditions at least that are very characteristic for interpreting the signals. The very first condition is that you have excitatory neurons that are basically going above the resting state activity and inhibitory neurons that go below. And what that does is feature analysis, effector driving as a mostly parallel LFV and mu ball changes. Sensory stimulation very reliably tells you that there is also a mu increase and there is an LFV increase. Then you have basically the opposite, but the opposite becomes immediately tricky. As you know, in cortex, in many structures, you have, in the cortex in particular, you have two ways to cause inhibition. The one is you go to the pyramidal cells and you tell them, shut up. And the other is you go and you activate the interneurons, and the interneurons tell to the pyramidal cells they should stop. So it makes a huge difference, because if you activate interneurons, you're going to have an increased bolt for suppressing activity. And if you do the first thing, you're going to have basically a decrease in the bolt activity that corresponds to a decrease in the neural activity. So the other thing that is very tricky and cannot be ignored for very long is the impulse response function that is a standard thing, the very first thing you do with system identification techniques if you want to understand enough a system and try to see how it is organized. This impulse response function in certain areas, like in cortex, is the way you would expect. You see it's here flat, and then it goes up. And the system is, in the mathematical term, is a causal system. You know that basically neurons are first, and vascular system is the second. And you measure the impulse response, the hemodynamic response of the vascular system, you expect to get this. But the thing that initially made me actually waste probably 2 weeks trying to see whether I have a bug in the software is that you have cases where there is activity before the 0 point. When you have an impulse response function with non-zero elements at the left of the trigger point, you have what people call anti-causal or non-causal system, which means there is some kind of feedback. And the feedback in this case is not surprising, because we know that the vascular system activity affects the astrocytes, the pericytes, and many other things, and it changes basically the story. So on top of it, and this is from thousands of data, I can trust them enough, there are characteristic differences, statistical significant differences between the impulse response function of different structures. So you can't generalize so much. The way you have maps that you use for whatever it is, either neuropsychological diseases or for making your ROIs or making whatever energy consumption, you need some ROIs that describe basically the activity of the system and the transfer functions between two different modes. So neural activity parameters they can be expressed as tensors of band-limited power signal, role of astrocytes, pericytes, capillary control, vascular constraints, and cellular vascular densities and spatial resolution of method. The last one here is a true problem if we ever want to understand very precisely, to know exactly what we are talking about, and we want to interpret correctly the functional imaging signals. And you can see here why. Because you have this is a series of studies that were done in the lab, and they will be continued in the future, because I do want to generate maps for at least the cortex in the beginning. Is you have a very, very, very complicated system, you can see here, and you have these penetrating arteries. The penetrating arteries, they kill laminar imaging. So you have to know, basically, if you want to have signals and you have the maps, you can avoid basically the areas that have the capillaries, the arteries that are penetrating, because they homogenize the differential, basically, activation of each one of the layers. So, and here you see the reconstruction, the complete 3D reconstruction is possible. We did it, it's published. And in the future, if we can do that for larger portions, we can generate maps that tell you basically you have your neural responses, then you have another basically phase where you have the vascular responses, and then you come up with a generation of the bolt signal. And you can do that inversely. Now, with all these things that I told you, what's happening with time? Oh, so I have 19 minutes? Okay, all right. I thought it's 19 seconds in the beginning. Okay, so Now, with all this, basically, we started a long time ago a large number of projects in different directions. First, to continue fanatically perception, learning, and memory. It's something that we are deeply interested. Many people, excellent people in the lab, they do this work, or they did it in the past. But then it's the functional neurovascular coupling, structural neurovascular coupling, molecular imaging. It's fantastic. One of the people that was in the committee, basically Goran Angelovsky, is the one who is the group leader in this fantastic project. They try to generate basically smart contrast agents that change the signal, instead of measuring basically things related to the blood. In cases that invasive procedures are allowed, you can just use the contrast agents. Then is interpretation of the neural signals, capillary microsampling, MRI with transsynaptic paramagnetic tracers, and so on. Today, in the rest of the time, I'll try very quickly to say a few words about the neural event triggered fMRI, and then tell you also how you can actually use this. Is the only thing that is relevant to the title. It's a way to see this as something approaching not the answer to the question, but the question itself. You know, if you want to answer what is a brain state, in order to ask the question, you need, before you ask the question, to know exactly how to ask the question. So, and now what we have, as I said, we do neurophysiology together with imaging. And neurophysiology, here you see basically signals from different structures that were not collected simultaneously. We can do it, but this is basically from different recordings, only because there are very characteristic events. And you can see here some up and down modulations. You can see somewhere here, you see this is basically the theta. You can see some other here, basically very strong deviations, deflections of the signal. And they're coming from different structures, and there are different events. And here is basically, this is the slow wave oscillation, the theta wave, spindles, ripple, K complexes, everything, PGO waves, you can imagine. Now you have that, and one of them, ripples, has been studied extensively. It was first reported in 1972 by O'Keefe, and then they put it in the 1978 basically book. They have the cognitive map, hippocampus as a cognitive map, they put it there. And it's the first description of what they called ripples, and later it was called sharp wave ripples, because you have this huge deflection. You would not be able to see the ripples unless you filter the signal in the frequency range. And then, of course, you see the low frequencies are out, and you see here basically the very fast oscillations. So Yuri Boutsaki, who is going to be here to give a talk in a couple of days, did fantastic work for 30 years on that. Most of the information we have is coming actually from his lab. And what we thought in the beginning, in trying a little bit to see how we can do something that correlates or associates basically local activity to something that you would call something like a metastate or whatever it is. So we thought to start with ripples, because the literature was extensive and we can get information basically understanding what the signal is supposed to be. So the ripple and all the other events are only interesting because it involves a huge network for their generation or their stopping or whatever it is, not a local thing. In particular, the brain normally, when you're active, right now when I'm giving my talk, my brain is dominated by theta oscillations, theta activity. So what happens, the theta activity involves a whole basically cascade of interactions. It starts in the nucleus reticularis pontine oralis, and from there it goes to the supramammillary nucleus, then it goes to the medial septum and diagonal band, as you know, of Broca. And then it goes to the hippocampus and a couple of other areas, and it pushes this kind of rhythm. An RPO actually has continuous. It starts basically for, I don't know, whatever, seconds, minutes, or hours. It starts firing like crazy, and this is converted into theta in the supramammillary nucleus, and then basically optimized in terms of synchrony in the MS, and from there it goes to hippocampus. Now, in order to have ripples, you have to stop that. If you stop it, then basically you see the ripples appearing everywhere. And Why did they draw attention and why did they put so much work? Well, there is more than enough indication that ripples, to some extent, they are not the only ones, but to some extent may be involved in the memory consolidation. As you know, the memory basically has two steps. The very first step is basically things go to cortex and they go also to hippocampus, but cortex is not learning very quickly, which is good news. And what happens is during resting state or during basically sleep and so on, you have activity from the hippocampus that retrains locally the tact areas and induces basically induction of LTP and so on and so forth. Now, it's indeed true that ripples coincide with the reactivation of neuronal ensembles that were excited during our waking learning experience. You all know the place cells. The rat is going around, and there are cells, they fire in a particular path, in a particular location. In the ripples, the same neurons that fired in a particular order, they fire during the sleep. So then the number of ripples increases after learning, and it predicts memory recall. Elimination of ripples during post-learning slow wave sleep interferes with memory consolidation, and ripples may also occur during the awake state at path choice points, potentially a quick recall mechanism. Now, what anybody would think is, okay, they cannot be and are not effectors. They are rather markers of brain states underlying memory consolidation. But if so, how can we see systematic up and down modulation patterns in a group of brain structures imaged with fMRI? And that is basically where we recorded from hippocampus about 8 years ago. And we recorded from hiccuppus, and also we did the imaging. And of course, we found immediately the ripples. And after we found the ripples, we detected the ripples. And after detecting the ripples, we took the fMRI signals with a huge help of Henri Evrard, one of the dear colleagues. And we had a very precise actual anatomical definition of regions of interest. And you see here how many signals you have. They can be from 56 to a very large number in cases that you use 2 hemispheres and so on. And what you do now, you have the events, and the events, you can take them, you can go to each event, and you have a peri-event trial, time before and after, and you can take these things and average them for each one of the regions of interest. Now, you have here, let's say as an example, this Royce, and here you have the time series that I told you. You see here time series, they show there is a reduction in certain regions of interest, and there is an increase. Here there is a massive reduction all over the place with other types of events. And from now on, you're not going to be seeing even this 3-dimensional surface, but you're going to be seeing actually just this here to make sure I can fit things within a page. And on top of it, you'll see also the activation fraction. By running basically different methods of selection of voxels, you can say that in the thalamus, basically that percent, 40% or 30% of the neurons they were, or of the region of interest was, reduced and down modulated, and 60% was up modulated. Okay, so far so good. Here's what you get. There are a lot of slides before we come to that, but we don't have the time. I apologize. So I'm showing you directly the results. What we got was really surprising. In the beginning, actually, I didn't even see that. Just we were random positions of the red and the black. But then when we sorted them according to the ontogenetic evolutionary pattern from basically brainstem to different metencephalon, mesencephalon, diencephalon, and so on. If you call them, then there is a clear story here. There is the subcortical areas are very strongly down modulated, and the cortical areas up modulated, with the exception of the foveal, basically region of the one for reasons that I may tell you later if we have time. Now, if you go in the record, you'll see that this time the description that you have with the spontaneous events of the expected neurophysiology are absolutely accurate. In the activated areas, they show increases in LFB and increases in the MUA. In the multi-unit activity, the drops, they show very, very sharp decrease in the multi-unit activity and so on and so forth. What is important is we put together basically the regions of interest to understand are there any systems in particular that have been suppressed. And this is indeed the case. The declarative memory system is the memory system that's being upmodulated. The procedural memory system is not the first time in the literature you can see with behavioral imaging or with patients or with functional MRI that they report often this antagonistic behavior between, let's say, the two types of memory systems. The thalamic nuclei are also down-modulated. The cholinergic system is completely down-modulated because, as you probably know, okay, I'll come to that. And the monoamine neuromodulatory system is down-modulated, and the forebrain system is also down-modulated, the neuromodulatory part. So, what are the interim conclusions? So ripples appear to activate cortical areas and not activate. The ripples occur while the cortical areas are activated, subcortical structures down-modulated. Down-modulated structures include thalamus, basal ganglia, cerebellum, pontine region, neuromodulatory centers. Ripple-triggered multi-structure activity, this is what this MSA means, in event type and structure specific, and state and species independent. So insights from concurrent recordings, up-down metabolism modulations fully reflect neural activity changes. Net BLP, if you take neural triggered BLP, basically time courses, points to antagonistic interactions between hippocampus and pons. I noticed that almost from the first minute because it was very striking. It was, fMRI has a variability. One of the most robust events is you see red hippocampus and blue in the whole pons. And so we thought we want to see what's happening. And indeed, as you all know, and you'll hear very nice things from Giulio Tononi and others, there is a systematic, basically, change in the states of the brain during sleep. You have the stages that people call basically the step 1, step 2, step 3 for the slow wave oscillations. And then you have the REM sleep, as you know, rapid eye movements, where you have all these very funny rapid eye movements. Now, the ripples were expected to occur only during the slow wave sleep. The PGO were expected to occur only during the REM, except that in a transition, small, at the level of a few seconds period, they may occur all together during this transition phase. Now, the two states are distinctly different neurochemically, except of basically the activity, the spiking activity, or all the LFBs. Hippocampal theta oscillations are very low during slow wave sleep. You have basic glutamate low and very high GABAergic because the reticular thalamic nucleus is actually doing a lot. Deactivating is causing basically this increase in the oscillations and deactivating the gamma range. Cholinergic activity low, dopamine low, and then norepinephrine and serotonin low with transient increases. So they may be involved in some ways in the memory consolidation as people speculated. The REM state, on the other hand, has almost the opposite. Hippocampal theta oscillations very high, not as high as during awakeness, but quite high. Then glutamate high, GABA low, cholinergic activity high, dopamine high, and so on. Now, the question, of course, is concurrent imaging with hippocampal, thalamic, and pons recordings. Can address the following questions. Are the ripple and pons event homeomorphic and homogeneous the way people thought? Are there event subclasses that may emerge together in either or both sleep states, simultaneously sequential emergence of ripples and PGO waves? Is there something like that? To cut it short, the answer is yes. What you see here is events in the LGN and in the pons. You see here LGN only, increases in LGN, for whatever reasons. You see here the true PGO waves that are increases in the pons and in the parabranchial nucleus and increases in the thalamus. And you see here only P waves, basically increases in the PGO. Now, if you see what the patterns are, the ones that are LGN, spontaneous increases, guess what they do? They activate basically visual structures. Whatever that may mean, I don't know yet, and I don't know when I know, but this is what's happening. Now, if you go to the other two types, you have basically the P waves, which is a mess. You don't get anything specific. And you have also the PGO waves that involve both increases of deflections in the pons and in the lateral geniculate body, and it is a mess. Now, that this here is a mess, of course, we never wanted to believe. We knew that most likely here you average basically different events. They may have completely different profiles. And are there PGO wave subgroups with distinct MSA profiles? And there are two types of PGO waves. And these two types of PGO waves, one of them is causing an increase in the MUA of the hippocampus, and the other is a very sharp decrease. If you see the profiles, here you see the ones that are basically SVR PGO and the ones that are only PGO. If you have basically the whole time series and you just look at your data, you'll see that in some cases, like the ones in the yellow, basically rectangle, you have the PGO waves occurring together with ripples. And in other cases, the PGO waves, they actually emerge alone. And if you see the MSA patterns, they are actually quite interesting. This is the one that I showed you previously with ripples, and this is the one you see with PGO. So there is an inversion, probably is basically a sequential hypothesis story where you have certain types of events that are responsible for having the tagging of synapses and then the LTP and the early genes and all the stuff that you need for long-term potentiation. And the results, the whole thing is, 3 minutes, okay. So Pontine Waves, there are two classes, and so what I told you, I'll just go ahead. Now, can multi-site recordings and MSA dynamics help us understand what a state is? Well, let's see. What I told you so far, you know already, there is a lot of neural signals. Right now, we can have up to 80 signals, 16 sites for 5 shanks, 16, 80 signals, electrophysiological from different areas. You have all the MRI signals, and you have also these patterns that we learn from the recordings that can kind of give us hints. We're not going to use them. The rest 3, 4 slides, they actually use only MRI to predict things, as if we didn't have physiology at all. So the question we ask is, can the MSA at every time point, the multi-structure activity, multi-ROI activity at every time point, be used for detecting neural events? As you realize, it's the only thing that we are doing that may even have very, very important medical clinical applications, because right now, if you want to find basically an epileptic site, you go with a barbecue method, the whole brain, you go to the temporal lobe, medial temporal lobe, and if you manage to interpret the patterns correctly and you have localization of MRI, you could find basically sources of oscillations without having the recordings at all. So, can these patterns be used for detecting neural events and potentially defining states? Well, let's see. Here you have the whole series that I show you repeatedly, and here you have the profile at each point. You see here that for each one of the points, you'll have a profile. I selected deliberately a profile here with random, basically, appearances of events. You see them. This is raw signal, MRI signal. And you have this pattern or you have this pattern. And this is from, a lot. It's 355,000 time points, a lot of sessions, a lot of experiments. So the statistics are very reliable. So you go here. And you see what I told you previously. You can see with your eyes that in the time series of the ROIs, well-defined ROIs, you can have disactivation patterns in the cortical and deactivation in subcortical region. And what you can do is you can have a shifted window like the one you see here, and go ahead and compute either the spatial PCA or take some kind of other statistics, Or you just low-pass filter the signal, which is absolutely fine. I've done both things, and they give the same results. If you low-pass filter the signal with a cutoff of, let's say, 75% of the Nyquist frequency of the signal, it's more than enough to get very good results. And here's what you get. Now, we got the signals. We use standard methodologies. They were developed by Michel Becherve and by Juan, and the student in the lab. They both actually somehow developed by some people that I don't even know of, but they were implemented by them in a very nice way. And by using these things, you can get basically from these thousands of MSA for each one of the time point, a clustering, and you can do it in a way, as I wrote it here, that you don't specify the number of clusters yourself. So there are methods like the normalized Katz index that make the algorithm to predict what are the optimal numbers of clusters. And in this case, you have 5 clusters. The one looks indeed just like the one I show you with Ripple, triggered basically MSA. The other looks like the Ponds. This one is a mixed thing, whatever it is, I don't know. This one, the same thing. And here's an activation that usually it's evident if you have very strong gamma activations in different structures. Now, this is basically the time course of these phases, each one of them. The index here is the index of the cluster. And you see basically there are alternations, short durations, longer durations, like what you would expect if you had other indices like the KH index and other things that tell you something about states. So here you see now the very first description. You see the 4 patterns that I show you. You see the profile of the patterns. You see the coherence map, like the ones that Gustavo was showing to you. And you see here basically this, if you concentrate on the lower half of this, you'll see that there is a red here and a blue here, which means actually there is antagonistic, the one's going up and the other's going down. The same thing here. In the gamma case, you have a global, basically, increases in coherence. This is a coherence map, basically phase differences in the signal. And the Ken-Harris index is something practical, but it's very nice. It gives you an idea of what could possibly be the state. And one means completely, basically, slow wave, sleep type of state. And 0 means basically activation of the gamma range and drop of the slow wave oscillations. So, question, what is the probability of occurrence of any intrinsic neural event during the prevalence of the MSA pattern? Is there anything? I must say this is going to be published, and not before too long. I was really pleasantly surprised. Look. This is each one of the clusters. In this first cluster, you're not using now physiology. You only use the physiology after you do the clustering, everything with MRI, and you take the physiology and you see basically in the hippocampus, the ripples, and the ripples occurring together with PGO, they have an increased probability of occurrence. The opposite happens with PGO. The spindles are also increased in the thalamus. The good thing with this methodology is you can use basically the fMRI from all sessions, although some of the sessions may have different recording sites. And with the PGO type of pattern, you have a very strong increase in the PGO probability of occurrence, and the same thing you see basically in the LGN and PGO, basically both of them together, they increase. And so that means that you have the chance actually indeed to localize, quote-unquote, neural events by analyzing the multidimensional MRI patterns. And this here is now what happens if you have electrical stimulation, just like the stimulation that Gustavo was talking about. Then the unsupervised and or without basically definition of number of clusters analysis, it shows that the states are reduced into these 3 from 5, and you have this mixing, whatever this may be. And then you have here this overall depression of at least metabolic activity, and here increase, and this increase, you see, this is here the direct electrical stimulation, and this is the visual stimulation. The reason it's messy is because after I saw what is happening, I went to old data, but I didn't have data that have random, this is what we'll do in the future, random stimulations, but they were visual and electrical stimulations. So you want it or not, you get them in the plot. So I'll skip it. This gives you also the coherence map. So I'm coming to the end. So sharp wave ripples and PGO waves have subclasses that yield fMRI MSA patterns which are specific to the level of their interaction and potentially to their type of event sequence, maybe Markov chains or whatever it is. The findings strongly suggest a reconsideration of the exact role of hippocampal and pons events and the dependency of their occurrence exclusively on brain states such as non-REM or slow-wave sleep periods. They also show that the slow-wave sleep and non-REM do not need, you're going to hear, I'm sure, from Giulio Tononi about this, does not to be global. It may be differentially appearing in different structures. And the robust and consistently observed inverse MSA patterns may reflect the time-order neuromodulatory fluctuations underlying synaptic, local plasticity, and system large-scale network-related memory consolidation. MSA can definitely be used for gaining insight into brain states. Now, here are the acknowledgements. I can't thank enough the people that you see here. The lab is, most of you know, is very large, has a lot of people. But first I present the people that have been directly working with me together, and they have huge contributions. And these are basically people related to methodology, with Axel being almost unique in his ability to make all the very complicated designs. And also here at the right, you see the scientists. And here you see the machine shops without actually the machine shops, the sophisticated machine shops, computer-aided designs, and so on. The whole project would be completely impossible to do. And I want to thank my current group leaders. They just we start also collaborate projects, and I want to thank basically all the people that have been in the lab basically and contributed in the project. Last but not least, I want cordially to thank the Max Planck Society, despite my unpleasant experiences with the current leadership, because I could not possibly do this research without the philosophy of the Max Planck Society. They are actually exceptional in providing true resources and permitting the existence of infrastructure personnel. You can be a person with 35 Einstein brains in your head. You are not going to be an electronic engineer and chemist and biologist and neuroscientist. You're not going to be this, right? So it's very important to have a group, and in most cases, as you know, it's very difficult to have a group. So the Max Planck Society makes all this possible, and I want to thank them. And thank you for listening to the talk.
Thank you so much, Nikos. As promised, fast, furious, stimulating, entertaining. Now I have to consult the coordinators if there is still time for questions. I see. Okay. So then with this clear sign, I am obliged to close the session, but I'm very sure that there will be many opportunities to ask all the questions you want to ask later this evening or on the following days. So thanks to everybody who has been here, and well, see you again tomorrow. Have a nice evening.